Hi everyone, we will continue exploring the topic Bioinformatics. In this session, we will continue to learn about a protein-ligand binding affinity model. This presentation is based largely on the information provided on the website, shown at the bottom of the slide. In session 8, we explored a protein-ligand binding affinity model using grid featureization. Back in 2023, when I first studied the book Deep Learning for Life Sciences, I successfully ran this binding affinity model based on the contents of the book. Fortunately, I captured a snapshot of output from that successful execution of the code. The results clearly demonstrate the Pearson's R-square values for both training and test datasets. It also shows how XY and weight data vectors look like from the training dataset for a successful execution of the code. When I run the same code now, I get Pearson's R-square values of nan and nan. Clearly something isn't right. To investigate, I compare the XY and weight data from both successful and failed runs. That's when I noticed an issue with the two-dimensional X dataset. Clearly the values are different. To pinpoint the source of error, I ran the same code on the Google Colab platform. This helped me to trace the problem back to the function load_pdbbind. It turns out that either the function source code has changed or the molnet pdbbind dataset has been corrupt. Since I was getting the empty array after loading the data with load_pdbbind. To work around this issue, we need to manually perform the key operations of load_pdbbind. This includes applying the featureization, in this case grid featureization, locating and loading the core dataset, and properly splitting the tasks, datasets, and transformer. Let's go ahead and manually do these operations step by step. Let's look into another protein-ligand binding affinity model. Prior to the script reading, let's first look into the coding and problem-solving strategy. Unlike the other textbook model, this web-based binding affinity model is working. However, it is not working as it is shown in the below website. The website is shown here. It is attributed to the fact that the sanitization part of the code is unable to filter out all the protein molecules that cannot be featureized by the context circular fingerprint function. It does filter out some, but not all. In the following example, it filters out 6 protein molecules instead of 42 protein molecules. Thus, I had to improvise and reorganize the working steps in the code to solve the problem of code crash. Okay, the working steps are as follows. First, we load a PDB files from the URL or protein databank website. Second, we already know that the information in the protein databank is produced by many scientists with limited facilities and resources, and thus the data may include errors or incomplete portion of the structure. Thus the PDB files need to be fixed, repaired, and cleaned. In the third step, after the sanitization step, the PDB files that failed sanitization need to be removed from the data or removed from the data set, since it will cause errors in the subsequent machine learning step. Now, the revised and reduced PDB file names are stored. Up to now, it is the steps exactly written in the code. However, if we continue proceeding with the code, errors will occur in the protein-ligand complex machine learning step. So, to solve this issue, I identified PDB files that failed to be featurized with the context circular fingerprint function. Then, I manually removed, one by one, these files from the downloaded dataset folders. This allows me to have the further reduced clean dataset of 151 protein-ligand data pairs. In the subsequent step, for the indexing purpose of the refined dataset, I reran loading PDB files from URL, which is step 1. Then I skipped step 2 and 3 and jumped to the step 4 to store the protein and complex PDB file names with the indexes. Then I continued to build the ligand-only machine learning model. And finally, I also built the protein-ligand complex machine learning model. To recap, the web-based model, the model in the website, includes the steps 1, 2, 3, 4, 8, and 9. But I had to add step 5, which is a manual removal step, and I had to repeat step 1 and 4 to make these machine learning models to work, to get the very similar results shown in the website. Okay, now let's look into the code predicting protein-ligand binding affinity. Unlike the previous model, you will see that this model is a bit more complicated, but it is working. What we do differently this time is that we will use circular and context circular fingerprints instead of grid featurization. Moreover, we will download the core PDB dataset from the protein data bank. As you know, we have already manually downloaded it from protein data bank when we worked on the code visualizing a protein, a ligand, and their complex. We will recycle the same lines of code to download the pre-processed data file that comes in the form of gzipped file pdb bind underscore core underscore df.csv.gz. First, we need to import the necessary modules such as deepcam, OS, NumPy, pandas, temp files, and cam under rdkits, or cam under rdkit.cam, and download underscore URL load underscore from underscore disk modules under deepcam.utils. Okay, second, we download the PDB bind data from the URL, and we get the current folder using get underscore data underscore dir function under dc.utils module. And then we join the current folder path and the file name to obtain the full file path. And then in the next segment, we will use exist function to script the if not structure, then do the following. First, we print the text, file does not exist, downloading file. Second, we actually download from URL using download underscore URL function, and the URL is shown in here. Okay, and then third, we print out the text file downloaded. And finally, the downloaded files in the full file path are loaded to the data frame. So this downloaded file in the full file path is loaded to the data frame called raw underscore dataset. This raw underscore dataset contains 197 rows and 7 columns, as we have seen it from protein visualizing example, but we will only actually use the information in the 3 columns, 3 column headings: PDB underscore ID, smiles, and labels, and produce the reduced size of data frame, which contains 197 rows and 3 columns. Okay, in the next step, we will download the .pdb files from the protein data bank and sanitize the structures. First, we import the necessary modules. We will import pdb file under open mm.app and pdb fixer under pdb fixer, and prepare underscore inputs under deepcam.utils.docking underscore utils, and we import time modules, and we create the array pdb IDs containing information under the heading of pdb underscore ID in raw underscore dataset. And we create another array ligand underscore smiles containing information under the headings of smile in the raw underscore dataset. Then we have the for loop structure. So for every protein and ligand in these 2 arrays, pdb IDs and ligand underscore smiles, we will do the following. First, we will download the pdb file corresponding to a protein labeled with the pdb ID from the protein data bank by using pdb fixer. We use pdb fixer function to download information regarding this protein ID, which is one of the protein in this dataset, pdb IDs. For every protein, we download it from this URL using this pdb fixer function and store it in the array called fixer. Then we write topology and position of the molecular systems to the corresponding file name. pdb ID is the file name, .pdb, this is the extension, and we will just write in this file. We will write this information into this file using write file function under pdb file. And then we will use prepare input function to prepare or sanitize the protein and ligand structure. Okay, this protein prepare input function has 6 arguments. The first argument takes the pdb file name, the protein file name, and then second argument takes the ligand structure, and the third argument replace underscore non-standard underscore residues equals true, indicates that any non-standard amino acid residues in the protein structure should be replaced with the standard amino acids. And then the fourth argument, remove underscore heterogenes equals to true, indicates that any non-protein or non-nucleic acid molecules in the protein structure, such as ions or cofactors, should be removed by these arguments. And then fifth argument, remove underscore water equals to true, indicates that water molecule in the protein structure should be removed. And the sixth argument, add underscore hydrogens equals to true, indicates that any missing hydrogen atoms in the protein and ligand structure should be added. And since not all the protein structure can be sanitized by the function prepare underscore input, it has try and except block to indicate the errors in the print statements. So when sanitization is failed, then it will print out the text, the ID numbers of protein, PDB ID, and failed sanitization. So we will know which protein files failed the sanitization. As I mentioned to you previously, it identified about 6 different protein structure that failed sanitization. Now, prepared protein and ligand molecules are converted to the molecular graph format and stored in protein ID P, the protein ID file, and then the ligand underscore protein ID PDB files. And this file conversion is for the further featureization steps in the next slide. In the next lines of code, we are selecting the files with specific conditions as the following. First, listdir function finds every file in the folder. In this case, it finds every file that has a character length of 8 and ending with .pdb, which is going to be the protein files. And all these names are contained in this set, and the set of protein file name are stored in the array proteins. Similarly, for the ligand files, the selection is made with the name starting with ligand and ending with .pdb. These file names are stored in the array ligands. These are the files including the ligand protein complex. In the next lines of code, we will handle the files with the failed sanitization. We find the files protein ID that is present in the protein file name but not in the complex file name, and then store these file names in the array called failures. Okay, -4 here is indicating that we will exclude the ending .pdb in the name set, and 7 indicates the ligands, excluding ligands and excluding .pdb. In the file name, we're only including the name of PDB ID. Basically, if I recap this line of code, it is actually excluding everything that has a pair, protein and the complex pair. And everything that doesn't have protein and ligand complex pair, all those names are going to be stored in the array failures. And then we find file name in this stored set failure, and then we will further remove these files from the data sets. When printing out this number of proteins and number of ligand sets data set, we will get 191 and 191, so which is 6 less than the original 197. So if we proceed with the code for the set of 191 data pairs, the code will not run completely. This is attributed to the fact that some data failed to be converted to the context circular fingerprints for some reason. Thus, I tested each of 191 data pairs and removed another 36 data pairs. Now, the remaining 151 data pairs are sanitized for both types of featureization. Thus, the same code is rerun after excluding the fixing portion of the code. Now we will rerun step 1, loading PDB file. Then we skip step 2, fixing portion of the code. Then we repeat part of step 3, storing the name to the array proteins and ligands. And then we will skip this sanitization part of the code. When printing out the remaining data pair, we get 151 and 151, which is 142 data pair less than the original data pair in this PDB bind core data sets. So further reduce data pairs. Okay, now we got our data pair ready. Now we work the core parts of the code. Featureization, fitting, model evaluation. The first model is a ligand-only model. First model is a ligand-only model, which relates the feature of the ligands to the binding affinity. Since the ligand is small, we can use one of the chemical fingerprint featureization techniques, which is circular fingerprints. And we're going to use the bit size of 2048. So there are 2048 features that's going to be digitized using this circular fingerprints. This extended connectivity circular fingerprints compute a bag-of-words style representation of a molecule by breaking it into local neighborhoods and hashing it into a bit vector of the specified size. And then we store this information into the array features. Then we create the full dataset using the NumPy dataset function, and X is going to be the features that have been featurized for every ligand in ligands, and Y is going to be the data labels, which is binding affinity values, and IDS equals PDB IDS, means that this is the index value for each protein ligand pair. Then we split the data set into two. Split random splitter dot train underscore test underscore split, and then we split the data set into train dataset and test dataset. Now that data set is assembled and organized in the format for the machine learning, so we proceed with the machine learning steps. Now we import modules for machine learning, random forest regressor, and we also import the evaluator under deepchem.utils.evaluate. We use the random seed of 42 as the random states to ensure the reproducibility of the random number generation during the model training. So C2, which goes in here for random states. As a machine learning model, we use random forest regressor of 100 decision trees, and the number of features to split will be the square root of the total number of features, SQRT. Then we use a fit function to train data. And then for Pearson correlation R-square value, we create a descriptor instance metric for Pearson R-square scores, and we evaluate the model with the training data set using the evaluator, compute underscore model underscore performance, and then we put the metrics in here, and then we print out the R-square values for training data sets. We do the same thing for the test data set, and then we print out the score. Okay? When performance metrics of R-square value is calculated, we get pretty high value for the training set, 0.935309. However, we get a pretty low value, 0.022125 for the test data set. This suggests the model works, and this ligand-only model may not be applicable to the real-world prediction. Okay, the second model is the protein-ligand complex model, which relates the features of the complex to the binding affinity. Since the crucial point of the protein-ligand complex is in near contact points of macromolecular complexes, we implement the contact circular fingerprints. We use the same bit size, 2048 features, digitized features. And as you can see in the descriptor instance, features we combine ligands and proteins. So here, instead of using just a ligand, we combine zip ligands, comma, protein. So we use both ligands and protein information, combine the information, and we do the featureization. So that's the only difference. And everything else is the same as the previous ligand-only prediction model. So The remaining code is basically the same as the ligand-only model. And when evaluating the model, we get lower R-square value, 0.398116. Okay, more complex structures, and then if we make the correlation with the binding affinity, even for the training set, we get the lower value than the ligand-only model. But then the test set has a slightly higher R-square values, but is still very low value, 0.025437. But these values are actually equivalent to the values that they indicated in the website shown below, which are comparable values. And these values suggest that the model works, but it may not be sufficient for the real-world binding affinity prediction yet. Okay, let's run the same code in the Jupyter Notebook platform. Since we have spent quite much time already going over the code, I will not go over the code again. The point to come across for executing this code is that the contact circular fingerprint function works with the further reduced dataset containing 151 protein and ligand pairs. However, since my last successful run on October 8, 2023, something in the contact circular fingerprint function has changed, that it generates more error as outputs. However, fortunately it still completes the run, which will be demonstrated after running all the cells of the code. First, let's have a look at the runs that have been already performed and saved. As you can see that it generates Pearson R-square values for the ligand-only model, and it also generated Pearson R-squares without any errors for the protein-ligand complex model with the contact circular fingerprints. But you will see that now it will generate some errors in this region as the outputs, but it will still complete the entire run. Okay, let's run the code by selecting Run All Cells. Okay, it imported all the modules, loaded the data, and imported more modules, and then it shows us that we are using 151 data set, and then we have some deprecation warning for the ligand-only model, but it still generated the output. The values are almost identical to the values shown in the presentation slide. And contact circular fingerprint, the protein-ligand complex model is still running. It will generate some error messages down here. This is what I meant. Error messages. Okay, the run just got completed, and it took some time to complete this run. And as I mentioned to you, since my last successful run on October 8, 2023, something in the contact circular fingerprint function has changed, that it generates more errors in the outputs. However, fortunately, it still completes the run, and the value that I obtained for the training set is 0.397981, which is very close to the value that I obtained last year in 2023, 0.98116. But the test set has a value of 0.011330, while my previous run showed me 0.025437. They're slightly different. This is the end of the session. I believe that we achieved several things throughout this session. First, we could successfully featurize the protein-ligand complex using the featurizer for the large molecule, contact circular fingerprint. Second, we identified the issue with the code execution, and it was mostly due to the PDB bind datasets. The sanitized and clean dataset for 151 protein ligand complex worked for the code. Lastly, the Python's open source code community code evolves. Thus, we need to learn how to troubleshoot the bugs or changes to get the code working. We have seen the same issue by learning about the Delaney's solubility prediction model. Without figuring out the correct syntax, we couldn't get this Delaney's solubility model working. So, in summary, we need to learn how to troubleshoot the bugs or changes to get this code working. Thank you for your attention. I hope you found the session engaging and informative.